Welcome back. This is mini lecture 3B. In this lecture, we're going to take a look at the structure and function of the nucleus, and we're also going to see how DNA is replicated. This slide illustrates the structures that you would find in a typical cell, specifically a typical eukaryotic cell. Surrounding the eukaryotic cell will be a cell membrane called a plasma membrane. This is a phospholipid bilayer with a bunch of embedded proteins in it. Its job is to surround the cytoplasm. And the cytoplasm is made up of a liquid or fluid-like component, I should say not a fluid, but an actual liquid fluid component called the cytosol, a protein scaffolding, which was referred to as the cytoskeleton. And because I'm talking about a eukaryotic cell, cytoplasmic organelles. And these organelles are little compartments within the eukaryotic cell that perform specific functions. They can be surrounded by a phospholipid bilayer, in which case they are called membrane-bound organelles, like the nucleus, the mitochondria, the endoplasmic reticulum. And then there are also non-membrane-bound organelles, which are referred to as ribosomes, cilia, flagella, and centrioles. During this course, we're going to learn a little bit about cellular anatomy, a little bit more on these cytoplasmic organelles and the cytoskeleton, but I wanted to introduce them to you now. Now, this is eukaryotic cellular anatomy. Prokaryotic cells will have a cell membrane. They will have a plasma membrane. Prokaryotic cells will have a cytosol. They will have somewhat of a cytoskeleton. It's not really well organized or well defined, but there is something of a support system. And then the big difference between eukaryotic and prokaryotic cells is that there are no cytoplasmic organelles in a prokaryotic cell. The nucleus is the organelle of eukaryotic cells where you're going to find most of the cell's genetic information. Most of the genes are found within the nucleus. Not all of them. This is because genetic information can also be found in the mitochondria and also can be found in chloroplasts in proteins. The nucleus is generally the most conspicuous, largest organelle that you will find in the eukaryotic cell. It is bound by two phospholipid membranes called the nuclear membrane or the nuclear envelope. And I like the term nuclear envelope better because if you think about the way a nuclear envelope is structured, if you think about the way an envelope is structured, it's made up of two pieces of paper, right? You have an outer sheet, you have an inner sheet, and then in between you could put the letter. It's the same idea with the nuclear envelope. There's an outer phospholipid membrane, there's an inner phospholipid membrane, and then there's a little space in between. In some cells, the nuclear envelope can connect to organelles like the endoplasmic reticulum, or it can connect to the plasma membrane. The nuclear envelope is not like a brick wall. It's not there to separate the inside of the nucleus from the outside of the nucleus. In fact, the nuclear envelope is designed so that there is import and export between the nucleus and the cytoplasm. So things can get in and out of the nucleus easily. And in order to do this, the nuclear envelope contains doorways or windows that are called nuclear pores. They are there for the exchange of materials between the nucleus and the cytoplasm. So they are there so that things like mRNA, messenger RNA, can exit the nucleus and enter into the cytoplasm. Ribosomal subunits can exit the nucleus and move into the cytoplasm, and cytoplasmic materials like proteins can get imported into the nucleus. Nuclear pores are large channels. They are made up of over 30 different proteins. These proteins belong to a family called nucleoporins. And on this slide, you can see I've got a picture of what these nuclear pore complexes or nuclear pores look like. They look like little flowers. And over here on the right-hand side, you can see, again, very simply, that the nuclear envelope contains these nuclear pores embedded in them. The nucleus is composed of the following three things: a nucleoplasm, a nucleolus, and chromatin. So if you look inside the nuclear envelope, you will find the nucleoplasm, the nucleolus, and DNA in the form of chromatin. We're going to take a look at each of these three component parts of a nucleus. The nucleoplasm is the specialized fluid you find inside the nucleus. It's very similar to the specialized fluid outside of the nucleus that we call the cytoplasm. The liquid portion of the nucleoplasm, the watery portion, is called the nucleosole. This is very, very similar to the liquid watery portion of the cytoplasm that we call the cytosol. So those two are very similar to one another. And just like the cytoplasm has a protein scaffolding network, so does the nucleoplasm. It contains a meshwork of protein filaments that we call the nuclear matrix or the nuclear lamina. Over here on the right-hand side, you see a picture of what these protein filaments can look like under the transmission electron microscope. The proteins that make up the nuclear lamina are varied and well described. There is a family of them that are called nuclear lamins. As a matter of fact, the majority of our nuclear lamina proteins are called nuclear lamins. And this nuclear lamina will function in a few specific ways. It will give the nucleus its shape, so it supports and shapes the nucleus. However, the nuclear lamina also organizes the DNA into what we call chromatin, and it also plays a role in replicating the DNA and transcribing that DNA into RNA. Nucleolus means little nucleus. It's a dense body of DNA, RNA, and protein that you'll find inside of the nucleus. It's not surrounded by any defining phospholipid bilayer membrane. So it's just a region where certain amounts of DNA, RNA, and protein are going to aggregate. If I say the word nucleolus, I want you to think ribosome subunit production. Inside the nucleolus, ribosomal RNA and ribosomal proteins are assembled to form the small ribosomal subunit and the large ribosomal subunit. We will talk a little bit later on about ribosomal rRNA, where it is and what it's for, but just for now, stick a pin in this. This ribosomal RNA is combined with specific ribosomal proteins to form the subunits of the ribosome. Once you make the small and the large ribosomal subunit, they migrate out of the nucleus via the nuclear pores into the cytoplasm, where they will combine with a piece of RNA called messenger RNA, and that assembled ribosome is then going to translate the messenger RNA into amino acids. That's a process called translation, and again, we're going to cover that in this course as well. If you're a cell that produces high amounts of proteins, like you're a highly synthetic cell like a neuron, your nucleolus is very, very prominent. Here you can see in this, on the right-hand side, this picture of a nucleus. It's a colorized transmission electron microscope picture, but I'm circling right here this very dense area of DNA, RNA, and protein. That's the nucleolus. The DNA found within a nucleus is found in a form called chromatin. And what chromatin is DNA helix loosely wrapped around proteins known as histones. Histones are only in eukaryotic cells. Therefore, the DNA inside of a nucleus is basically only found in eukaryotic cells. Now, I'm not saying that the DNA inside of a bacterial cell cannot be organized into a chromatin-like structure. It is. Okay, prokaryotic cells don't have nuclei, but they do set aside a region of their nuclei, a region of their cytoplasm where their DNA is found. And in that region of the cytoplasm where you find the DNA, you can find histone-like proteins that help organize the bacterial DNA into something that looks like chromatin, but it's not. Chromatin, histones, these are eukaryotic things. The functions of chromatin are shown here. The fact that the nucleus is a really small organelle means that we have to have a way of packing our DNA into a really, really small volume so that it can not only fit in the cell, but fit in the nucleus of the cell. In a human cell, there are 46 chromosomes in the nucleus. If you took the DNA helices that make up all 46 of those chromosomes, strung them end to end, you would have over 2 meters of DNA helix. The typical nucleus of a eukaryotic cell is maybe 10 microns in diameter. So that's a massive packing ratio. we've got to take that really, really long 2 meters of DNA and pack it into something that's only a few microns in diameter. That's the job of chromatin, to package DNA into a really small volume. I don't know if you guys have ever seen those commercials about space saver bags, how you see those people who take like 50 sweaters and they put it into this bag and then they suck all the air out of the bag and the bag shrinks down to this little tiny flat thing. That's what I think of when I think of how chromosomes are packaged into, how chromatin packages the DNA helix into a small volume. The fact that you've also organized it around these histones, you've strengthened the DNA. The DNA helix is very, very delicate. So if you can wrap it around these histones, you give it a little bit of support and a little bit of strength. And the fact, we're going to see that you can actually organize chromatin into multiple levels. And this allows us actually to separate our DNA so that we can separate it into daughter cells. This strengthening also prevents DNA damage. And then finally, the association of DNA with a histone imparts a certain amount of regulatory influence. We won't get into this, but I'll tell you that when DNA is wrapped around a histone, it becomes inaccessible to the machines of DNA replication and the machines of transcription. And in order to do certain things with DNA, you have to slide those histones out of the way. So the fact is that chromatin imparts a certain level of regulatory control over what you can do with the DNA. Chromatin is often described as, quote unquote, beads on a string. So the word beads on a string model is frequently used to describe chromatin. And the reason why it has that term is that if you look at it under the microscope at a very, very high magnification, you can actually see these little round beads, and you can see the DNA kind of wrapping around it. These beads we're going to see are called histones, and the DNA wraps about 2 times around this histone bead to form a structure called a nucleosome. And this nucleosome I have highlighted here with this red box. The DNA helix, which is found in between adjacent nucleosomes, is called linker DNA. I'm going to box in what the linker DNA is right here. So here's linker DNA. Here in this picture I've drawn 3 nucleosomes all in a row, and the DNA helix that links these 3 nucleosomes is called linker DNA. The amount of linker DNA between nucleosomes depends on the species. That's about 50 base pairs in human DNA. It might be a little longer or a little shorter in another species. What I've diagrammed right here is a type of chromatin called the 10 nanometer fiber, or euchromatin. Before we continue on learning a little bit about chromatin, let's take a step back, because I mentioned that the beads on a string model, the bead is the histone. So what exactly is a histone? It's a complex of 8 small proteins, each of which have an overall positive charge. The histone is said to be an octamer because it is made up of those 8 small proteins. Specifically, a histone is made up of 4 pairs of proteins. There are 2 histone proteins called H2A, 2 proteins called H2B, 2 proteins known as H3, 2 proteins known as H4. So 2, 2, 2, and 2. You've got 8 proteins which come together. The DNA is then going to wrap around this histone octamer core about twice, and the distance or the diameter across this histone bead is about 10 nanometers, which is why the beads on a string model euchromatin is referred to as a 10 nanometer fiber. There are 2 other histone proteins that are called H1 and H5. These are not part of the histone octamer. Instead, they're called linker histones. There isn't a lot known about H5, but there's a little bit more known about H1. When the DNA helix wraps around the histone bead, H1, which you can see here as this green histone, will then come in and lock it into position so that the DNA helix doesn't slip. So this is the bead and the beads on a string model. 10 nanometers in diameter. This is why we often call euchromatin the 10 nanometer fiber. If you take these H1 proteins and you interact them with one another, you're going to get the next level of chromatin organization, which is 30 nanometers wide, which is known as the heterochromatin fiber. Histones are positively charged. That octamer of 8 histone proteins, each one of them have a positive charge. They're made up of positively charged amino acids. So the overall histone bead, positively charged. This means that the proteins of the histone form simple ionic bonds with negatively charged DNA. There is no fancy way that DNA and histones interact with each other. It's not driven by DNA sequence. It's not driven by anything that is chemically complicated. It's just simply the fact that the histone is positively charged and the DNA is negatively charged. However, you can alter this positive-negative interaction by chemically modifying the histone or chemically modifying the DNA. For example, if you take acetyl groups and add them onto those histone proteins, this is going to lessen how positive the histone is, and it's going to loosen the association between DNA and its histone. So acetylation of histones loosens the association between DNA and histones. If you were to add methyl groups to these histone proteins, it actually makes them more positive and it tightens the association between DNA and the histone. So methylation of histone proteins tightens their association. Methylation of DNA sequences can also tighten the association between DNA and its histones. And the reason why this is important is because the tighter you wrap the DNA around a histone, the less accessible it is. In order for things like the DNA replication machinery or the RNA transcription machinery to do their job, they have to bind to DNA and move along it. And that's going to be really, really hard if that DNA is attached to a histone. So in order for DNA replication and RNA transcription to happen, we have to loosen the association between DNA and its histones. So we have to acetylate the histones before we can do things like replicate the DNA and transcribe it into RNA. The minute we're done with the replication and the transcription, we want to tighten that association up, so we remove the acetyl groups and we add in methyl groups. And methyl groups makes that association just that much tighter. Because the histone bead is 10 nanometers in diameter, the DNA that is wrapped around these histone beads is called euchromatin. Euchromatin is the loosest form of chromatin that we have in the cell nucleus. Now you can pack this 10 nanometer euchromatin fiber even further by allowing the H1 histone proteins to interact with one another. That produces a fiber which is 30 nanometers in diameter or width, and it's called the 30 nanometer fiber or heterochromatin. It's kind of a big solenoid structure, so it looks almost like a telephone cord, and it's shown right here, right at this part of the figure right here. You can see how it looks almost like a telephone cord. That is the 30 nanometer fiber. You can then take this heterochromatin and you can compact it and loop it and organize it even further. You can form these loops that actually will attach to a bunch of nuclear proteins, loop it again, loop it again, loop it again, and you form almost like a flower-like structure. These structures are 300 nanometers in length. So from one flower petal to the next flower petal on the opposite side, that's 300 nanometers. And if you take a bunch of these flowers and sort of all line them up on top of one another, you get basically the chromatid chromosome. When the cell is not dividing, in other words, when the cell is in interphase, the two forms of DNA you will find are euchromatin and heterochromatin. As the cell goes through the cell cycle, as it goes through interphase and it starts to approach the M phase, you are going to take all of that euchromatin, you are going to duplicate it, and then you're going to condense it from euchromatin to heterochromatin to that flower-like structure that's the 300 nanometer fiber, and then stack all of those flowers up on top of one another to form the chromatid chromosome. And what you end up with is a super condensed duplicated chromosome with two sister chromatids joined at the centromere. The condensing of chromatin through its various forms is regulated by specific proteins. So for example, going from euchromatin to heterochromatin is regulated by the H1 histone protein. Going from 30 nanometer fiber to the 300 nanometer fiber is regulated by proteins of the nuclear lamina. And one of those proteins in the nuclear lamina that is responsible for condensing to the duplicated chromosome form is called condensin. Molecular and cell biologists are not inventive people when it comes to naming things. So when they found a protein which was critical for condensing DNA to a duplicated chromosome form, they called it condensin. Chromatin arrangement is critical to the function of DNA. I want you to get this as a take-home message. The more condensed your chromatin is, the less accessible it will be to molecular machines that are responsible for DNA replication and gene expression. So if you can't get to that DNA helix because it is condensed and compacted, you can't do anything with that DNA helix, like replicate it or transcribe it into RNA. We're now going to take a look at how DNA gets replicated. And before we move on to DNA replication, I want to remind you what DNA is made out of. First of all, DNA together with RNA, these are known as our nucleic acids. Nucleic acids are made using carbon, hydrogen, oxygen, nitrogen, and phosphorus atoms. The building blocks of DNA and RNA are known as nucleotides. Nucleotide is made up of 3 component parts. It's made up of a 5-carbon sugar known as a pentose sugar. Attached to the 5th carbon of that pentose sugar is either 1, 2, or 3 phosphate groups. You can see here in this picture of a nucleotide that there's only 1 phosphate group shown here. And attached to the 1st carbon of the pentose sugar is what is known as a nitrogenous base or nitrogen-containing base. This base is also referred to as an organic base. In terms of DNA and RNA, there are 5 types of organic bases you will find. You will find either adenine, cytosine, guanine, thymine, or uracil. Both RNA and DNA will have adenine, cytosine, and guanine. DNA will have thymine, and uracil will be found associated with RNA. The sugar and the base collectively are known as the nucleoside, and then to this nucleoside you attach either 1, 2, or 3 phosphate groups. So DNA, deoxyribonucleic acid, is made up of DNA nucleotides, and RNA, ribonucleic acid, is made up of RNA nucleotides. Just to review again that there are 2 families of nitrogenous bases. Cytosine, thymine, and uracil are known as the pyrimidines. Adenine and guanine are known as the purines. There are 2 kinds of pentose sugars that we find in nucleic acids, and these are called ribose or deoxyribose. And the cool thing about these 2 sugars is that they are identical except for the 2nd carbon. If you take a look at the 2nd carbon in deoxyribose, you see we have a hydrogen. And if you take a look at the 2nd carbon at ribose, you see we have a hydroxyl group. So the only difference really is the lack of an oxygen at the 2' carbon. Hence, this is why we call DNA deoxyribonucleic acid, because it is missing that oxygen at the 2' carbon. Nucleotides in the nucleic acid are linked together to form a polymer called a polynucleotide chain. So in other words, a nucleic acid can be referred to as a polynucleotide chain. To build a polynucleotide chain or a nucleic acid, what you do is you link nucleotides together. And the bond that forms between adjacent nucleotides is referred to as a phosphodiester bond. Phosphodiester bond will form between the 3' hydroxyl of the nucleic acid chain and the 5' phosphate of an incoming nucleotide that is being added to that chain. The exposed phosphate group found at one end of the polynucleotide chain is referred to as the 5' end, and the exposed 3' hydroxyl found at the other end of the chain is called the 3' end. So there is directionality to a polynucleotide chain. As I alluded to in the previous slide, it is only this 3' end here, this 3' hydroxyl, that is capable of forming a phosphodiester bond with an incoming new nucleotide. You cannot add a nucleotide to this 5' phosphate. The enzymes that make these phosphodiester bonds can't do this. Instead, the nucleotide gets added to the 3' end of the chain. So again, think of this 3' hydroxyl as being the reactive end of the polynucleotide chain. Because of this directionality, we refer to the DNA or RNA chain growing in one direction only. It grows from 5' to 3'. DNA is actually two DNA polynucleotide chains. They are oriented in opposite directions so that the 5' end on one chain is going to be at the opposite end of the 5' end of the second chain. The sugars and the phosphates will form basically the backbone, and the bases, the nitrogenous containing bases, face inward to that double helix. So think of the DNA helix as being a spiral staircase, with the railings being the sugars and the phosphates, and the steps being the bases. You'll note that these bases pair up in a very specific way. They are known as complementary base pairing, and these complementary bases are linked by hydrogen bonds. The search for the structure of DNA started in the 1940s, and there were several groups scattered between the United States and Europe that worked on figuring out just exactly how the DNA strand became the DNA helix and how this DNA helix was structured. Three of the more well-known researchers in this area were Rosalind Franklin, James Watson, and Francis Crick. And Rosalind Franklin worked in one laboratory and Watson and Crick worked in another one, and they both worked in England, again, in the late 40s, early 1950s. So they were competitive labs in trying to figure out the structure of the DNA helix. It was Rosalind Franklin, actually, that took a very famous photograph of DNA, and she began to propose the structure of the DNA helix. Her work was instrumental in James Watson and Francis Crick being able to publish their data in 1953, and ultimately for them to win the Nobel Prize. There is quite a controversy associated with Rosalind Franklin, James Watson, and Francis Crick. So if you're interested, you could actually Google these three researchers and find out essentially that Watson and Crick basically stole Rosalind Franklin's work. Beyond that, I can just tell you that through their work, we discovered that the bases of the DNA helix face inward, and that the sugars and phosphates form a sugar-phosphate backbone, and they're found on the outside of the helix. So you can think of the DNA helix as being a spiral staircase in which the bases are the steps found inside, and the sugars and the phosphates form the railings. Again, this work done by these researchers led to the awarding of the Nobel Prize to Watson and Crick and to Maurice Wilkins in 1962. Maurice Wilkins actually was a colleague of Rosalind Franklin. I believe he was her boss at the time. Rosalind Franklin died before the Nobel Prize was awarded, and Nobel Prizes are not awarded posthumously. So that was why she was never named on the Nobel Prize for the work, honestly, that she spearheaded. Through the research of groups that were led by Rosalind Franklin and Watson and Crick, proposals were made about the DNA double helix. One proposal was made proposing that the two DNA strands in the helix are held together by hydrogen bonds found between the bases. And these are shown as these little dotted red lines right here. Now these bonds, hydrogen bonds, are not very strong. So the DNA helix can be easily separated by extreme conditions like high heat, or extremes in salt concentration, extremes in pH, and even mechanical forces. The loss of the natural state of a molecule is called denaturing. So separating the DNA helix into two separate DNA strands is called denaturation. Erwin Chargaff, who was another researcher in the DNA field, proposed that these bases pair in a complementary fashion. And he proposed what was the rule, which is a purine base pairs with a pyrimidine. And this kind of makes sense because purines are large, if two purines base paired with one another, you'd get a bulge in the DNA helix. And if two pyrimidines base paired with each other, the DNA helix would have a dent. And we know through Rosalind Franklin's work that this doesn't exist, that the diameter of a DNA helix is consistent throughout. The rule is an adenine base pairs with a thymine and two hydrogen bonds hold those together. And the other rule is that a guanine base pairs with a cytosine and three hydrogen bonds hold those together. Now, by virtue of three hydrogen bonds, that means a GC pairing is stronger than an AT pairing. So if you have a DNA region which has a lot of G's and C's base pairing, then that region of the DNA helix is going to be harder to denature versus a DNA region that has a lot of A and T base pairs. When we talk about the sequence of a DNA strand, we kind of short form it. We just use the names of the bases. So if in other words, we don't use the nucleotide names because they're really long. So instead of saying the sequence is deoxyadenosine triphosphate, deoxycytosine triphosphate, deoxyguanosine triphosphate, we just say ACG. DNA is a double helix of two DNA sequences that are complementary to one another. Remember, the DNA helix is made up of two DNA strands that run anti-parallel. Remember, bases are complementary, so our two DNA sequences are complementary to each other. To simplify things, we call one strand the sense strand, we call the other one the antisense. Now, in nature, there's no such thing as sense and antisense. The cell doesn't look at two DNA strands and goes, oh, this one's the sense and this one's the antisense. But to define them, the antisense strand is the template strand we use in transcription to create mRNA, and the sense strand is the other one. If you look at the sequence of a sense strand, it has the exact same DNA sequence as the mRNA strand, except in the DNA, we're going to have T's, and the mRNA, we're going to have U's. Now, this doesn't make any sense right now, but don't worry, we're going to go over a lecture of transcription, so it will. So for now, the antisense strand is what we use to transcribe into mRNA, and the sense strand is the opposite. When we write out our DNA sequence, the sense strand is at the top with the 5' phosphate on the left and the 3' hydroxyl on the right. The antisense is written on the bottom and it's flipped. So its 5' end is on the right and its 3' end is on the left. The double-stranded DNA helix has to be unwound before we can replicate it. And it's unwound into individual strands that are called template strands. An enzyme called a DNA polymerase is going to attach to each template strand, and it's going to move along each template strand and create a complementary sequence of DNA that we call daughter DNA. So in this picture here, we have the template strand, which is in purple, being unwound in this direction. We now have two purple strands called the template strands. The DNA polymerase is going to attach, and it's going to work its way down the template strand, reading the bases and bringing in the complementary nucleotide. What it does actually is it brings in the complementary nucleotide. It attaches it to the base first, so it's going to attach it, for example, to the C, and then it's going to form a phosphodiester bond so that you now link it to the growing DNA daughter strand. So who are the players in this process we call DNA replication? Well, the star of the show is called DNA polymerase 3. This is the polymerase that's going to read the parental DNA template strand. It's going to bring in the complementary nucleotides and string them together through phosphodiester bonds and through hydrogen bonds to the parental template so that you make a daughter strand. But we have to unwind this DNA into two individual template strands. That's done by an enzyme complex called helicase. It is said to either melt or unzip the double-stranded parental helix into those two template strands. They're going to be single-stranded binding proteins that are going to hold the unwound parental template strands away from each other so that they don't zip back together and reform the helix. There's going to be an RNA polymerase complex called primase, which is going to create a small RNA oligo or a small RNA primer, which is going to complementary base pair onto the parental DNA template, and it's going to give something for the DNA polymerase 3 to attach to and extend off of to make that daughter DNA strand. And finally, we're going to have an enzyme complex called DNA ligase that's going to basically fill in any of the missing phosphodiester bonds in the daughter DNA strands so that the daughter DNA is one long continuous piece. This is actually going to be the bacterial mechanism of replication. It's a little bit more simple. The eukaryotic way of replicating DNA is the same overall mechanism. It's just that there's more players here. So that's why I like to teach the bacterial mode of replication, because there's just less moving parts to talk about. You've seen the word polymerase. What is it? Well, I see the end of that word as being ASE, so I know that it's an enzyme. And I know that it's an enzyme that's going to make a polymer. So if you are a DNA polymerase, you are going to make a polymer of DNA. If you're an RNA polymerase, you are going to make a polymer of RNA. And what these polymerases do is they create the phosphodiester bonds between adjacent nucleotides. In DNA polymerase, they also create the hydrogen bonds so that the daughter DNA strand can base pair with the DNA parental template. Now in bacteria, there are 3 DNA polymerases. The one that we really are interested in, the star of the show, so to speak, is called DNA polymerase 3. Well, if there's 3 DNA polymerases, you know there's got to be a DNA polymerase 1 and a DNA polymerase 2. DNA polymerase 1 is part of the replication machine. It is going to remove any RNA nucleotides or any incorrect nucleotides that it sees and fill in the gaps. It's removing of incorrect nucleotides and correcting them and putting the right one in. We call this proofreading. DNA polymerase 2 is also thought to be involved in proofreading. Now, the reason why we're doing this is that there's basically 3 DNA polymerases in bacterial replication. There's something like 11 polymerase subunits that you have to talk about when you talk about eukaryotic cells. So it's much easier to talk about the DNA replication mechanism in bacteria because it's just, like I said, it's got less moving parts. DNA replication in bacteria is performed by 3 DNA polymerases called DNA polymerase 1, 2, and 3. DNA replication in eukaryotic cells is performed by similar DNA polymerases, but we have a little bit more. We have something like 5 or 6 polymerases. So to make things a little bit more simpler, I'm going to tell you about the bacterial DNA replication mechanism, and I'm going to tell you about DNA polymerase 1, 2, and 3. The star of the show is DNA polymerase 3. DNA polymerase 3 is the actual protein complex that's going to read the DNA template and string together a bunch of daughter nucleotides to create the daughter DNA strand. DNA polymerase 3, as you can see in the picture here, is a complex of multiple protein subunits that all work together in a coordinated fashion. The core of the DNA polymerase 3 is this large orange one that I'm going to box right here in red. This is where the daughter nucleotides are strung together. So it contains the active site for nucleotide addition. It is the heart of the polymerase. There are a couple other subunits I want you to know. For example, I want you to know down here this sigma epsilon subunit. This is a partnership that reads the DNA, daughter DNA nucleotides that are being brought in by the alpha subunit. And if they're correct, great. If an incorrect nucleotide is brought in, it stops the polymerase immediately and corrects it. We call that proofreading. When an enzyme involved in replication looks at the daughter DNA strand and double checks to see if all the nucleotides are correct, that's called proofreading. So the epsilon sigma subunit partnership is responsible for proofreading the addition of the nucleotide immediately. Here's the thing though, it can't go back and correct a mistake it made 2 or 3 nucleotides ago. So it has to correct the mistake the minute it makes it. The other one I want to show you here is the beta clamp right here. I'm going to circle it right here now in blue. This beta clamp is a donut-like clamp that clamps around the parental DNA. So it actually clamps the alpha subunit, the sigma and the epsilon subunit. It clamps the DNA polymerase III complex to the parental DNA template. I'm going to tell you about this purple one in a second, but I will then highlight all of these other Greek letters here: delta, delta prime, gamma. I think this one is phi, this X is chi. All of these extra subunits, the ones that are not the beta, the epsilon, the sigma, the alpha, all of the extra subunits work collectively to be called what is called a clamp loading protein. This is the part of the DNA polymerase III that will take the beta clamp on and off. So it will open the beta clamp so that the DNA polymerase III complex can come off the DNA template, and then it'll clamp it back onto the DNA template. The last thing I want to tell you about is this purple subunit called tau. And you can see it is sort of dual-sided. And if you can see in this diagram, it is actually linking two DNA polymerase III complexes to one another. And that's what I want to emphasize in this slide. It's two DNA polymerase III complexes that coordinate with each other, and they replicate both DNA templates at the same time. Now, this is going to be easy for one of the DNA templates because it's going to be in the correct orientation. But now I'm going to tell you in the remaining slides about how the DNA polymerase III complex overcomes the fact that the other DNA template is in the complete opposite orientation. DNA replication doesn't just start at any random sequence of DNA. It begins at a specific sequence of DNA called an origin of replication. In bacteria, it's called ORIC. O-R-I-C. And because bacterial chromosomes, there tends to be only one, and it's a nice big circular piece of double-stranded DNA, they have one origin of replication. But in eukaryotic cells, as we became more complex, we developed more chromosomes. We have 46 chromosomes in our cells that we have to replicate. So there are going to be multiple origins of replication found along those eukaryotic chromosomes. What is the origin of replication for? Well, it's for the binding of what I call the replication machine. It is a large complex of multiple proteins that all work together to replicate the DNA. And this replication machine is going to recognize the sequence called the ORI sequence, the ORIC sequence, and it's going to bind there. As a matter of fact, you're going to have two. You're going to have one replication machine that'll bind on this side of the origin. You'll have another replication machine that'll bind here. On this origin, we put a replication machine on the left side. We put one here on the right side. And I'll do the same for this origin of replication. These blue circles that I just drew, think of them as the replication machine. It's going to contain things like the DNA polymerase III. Like I said, they're going to recognize the sequence called ORIC. They're going to bind one on one side, one on the other side, and they're going to head off in opposite directions to each other. And all of those all of those replication machines are going to do that. They're going to head off down their DNA strands, replicating their DNA strands, and eventually they're going to meet another replication machine coming from the opposite direction. When they do, they kind of bang into each other and fall off of the DNA, and DNA replication is said to be complete. DNA replication can be quite complex, and this is because there are two DNA templates we have to duplicate. We have to duplicate them at the exact same time. One of them is in the perfect orientation to allow for easy, efficient replication. The second DNA template is not. It's in the complete opposite orientation, making replicating that template strand much more difficult. So over the years, I've developed an analogy, and my analogy is zip lining. So I say DNA replication is kind of like going zip lining. Here's you and here's your partner. You are zipping down the zip line, having a lot of fun, but let's use that as our analogy for DNA replication. The DNA polymerase III alpha subunit is either you or your zip lining partner. And again, you are going down these two zip lines at the exact same time, so you are physically tethered to each other. I'm going to put a little belt here that physically attaches you and your partner together. The DNA polymerase beta clamp, that's the carabiner harness that attaches you to the zip line. The clamp loading protein, well, that's the zip lining employee that will clamp you onto the zip line and take you off. The RNA primer is, think of it as a wooden platform that you stand on as you're being attached onto the zip line. So while it's not there, I'll draw like a little box right here. So that's your wooden platform that you stand on, and then the zip lining employee hooks your harness up to the zip line. And then I can even extend the analogy out even further to say that, you remember DNA replication only begins at specific sequences called origins of replication? Well, consider the origin of replication to be the parking lot that you pull into before you go ziplining. The DNA replication complex, or what I call a DNA replication machine, is a large aggregation of protein complexes that come together and work together to replicate DNA. And as I mentioned in the previous slide, they recognize the sequence called the ORI sequence, the origin of replication, and two replication machines are going to bind, one on either side of that sequence. What's found in a DNA replication complex? A bunch of things. But here's what I want you to know. It contains a helicase to unwind the two DNA template strands, a primase, which is going to create a small RNA primer for the DNA polymerase complex to bind onto, two DNA3 polymerase complexes, which are going to be bound to each other by the tau protein, and then all those other proteins, which collectively I refer to as the clamp loading protein. There are some problems with replication that we need to overcome. The first problem is that DNA is found in the form of chromatin, yes, but DNA associated with chromatin can supercoil. So the minute you loosen up the DNA, slide the histones out of the way, you've created this big long stretch of DNA that is free to be replicated. But because it's physically attached to things like histones and physically attached to, in bacteria, cytoskeletal proteins, the minute you start to unwind that DNA through a helicase, you are going to create knots ahead of that unwinding, and that's called supercoiling. So in both prokaryotic and eukaryotic DNA, the DNA is physically attached to supportive proteins, be it histones, or nuclear lamina proteins, or the supportive proteins of a prokaryotic cytoplasm. And because of the second problem, the DNA helix being a helix and us having to have a helicase and unwind the DNA, we're going to create tension ahead of that unwinding. That's called supercoiling. There are enzymes to get rid of these knots, these supercoils. They are called topoisomerases. They nick one of the two strands, so they kind of cut one phosphodiester bond in one of the two strands of the DNA helix. That strand can then whip around, relieve the tension, you get rid of the supercoiled knot, and then you connect it back together again. So as our helicase is unwinding a section of DNA, there is a topoisomerase that is getting rid of the knots that this unwinding is creating, the supercoils that this unwinding is creating. So as that helicase moves, there's got to be a topoisomerase in front of it in order to get rid of the supercoils. So the first two problems are kind of linked together. The third problem on this slide is that the minute you unwind a DNA helix up into two template strands, they are going to want to rewind back together again. So there are single-stranded binding proteins that physically bind the single-stranded templates and prevent them from complementary base pairing up with one another. It's kind of like, I say this to the girls in the class, it's kind of like when your friend breaks up with her boyfriend and she wants to call him and you take her cell phone away from him. Or her, right? Guys would never do that, right? Guys, you never do that, do you? Like I said, it's like you take your cell phone away from your girlfriend and say, no, you can't call him. Guys are like, yeah, you want to call your ex-girlfriend? Go for it. Now you're all laughing right now because guess what? You know this is true. Okay, we fixed a bunch of problems. We have figured out how to unwind the DNA helix using a helicase. We've gotten rid of any knots that we create, any supercoils that we create because of this unwinding. That's done through a topoisomerase. We have figured out how to hold the two parental DNA templates away from one another using single-stranded binding proteins. The next problem is that DNA polymerases, which are part of that replication machine, they can't bind single-stranded proteins. sorry, they can't bind single-stranded nucleic acids. So we've got these two single strands of DNA, and the DNA polymerase III can't bind it. In other words, the unwinding gets rid of the base pairing that our DNA polymerases need. Well, we're going to solve this problem using an enzyme called a primase. A primase is going to make a small piece of RNA. It's going to be called a primer. It's going to bind to the DNA strand, and it's going to create a little double-stranded hybrid of DNA and RNA. And now our DNA polymerase III will see that piece of double-stranded DNA RNA and go, aha, that's where I bind. Now, I don't know why it's RNA. I don't know why it's an RNA primase that makes a small piece of RNA. I just know that that primer is made up of RNA. And now that we have this piece of double-stranded DNA RNA, the DNA polymerase III, which is part of the replication machine, can come bind to the 3' end and head its way down towards the 5' end of the template strand. So the way, continuing my ziplining analogy, the way I look at the primase and the primer is that the primer is a little wooden platform that you, as the alpha subunit, is going to climb up onto. On that primer, there will be the zipline worker who will take the big beta clamp, the big carabiner, and attach you onto the DNA template. And then you can zip down, heading down to the 5' end of your zip line. The antisense strand will have one of these platforms, one of these primers, every 100 or 200,000 base pairs. That's because the antisense, I'm going to tell you right now, is in the correct orientation for easy, fast, efficient DNA replication. The problem is your partner that you are tethered to, the other DNA polymerase III, well, its DNA template, or its zip line in our analogy, is in the wrong orientation. Now I'm going to show you in the next slide how that gets fixed, but for now I'm going to say that the sense strand, your partner's strand, which is in the wrong orientation, you're going to have primers being made every 1,500 to 2,000 base pairs. So you're attached to the antisense strand. You need a primer or one of those wooden platforms that you're going to climb up onto every 1 to 200,000 base pairs. Your partner is going to have those wooden platforms, those primers built every 1,500 to 2,000 base pairs. And we're going to see why in the next slide. The biggest problem in DNA replication is that the two parental strands in the DNA helix are opposite in their chemical polarity. And as I've said before, we're replicating both of these DNA parental template strands at the same time. We have two DNA polymerase III complexes that are physically tethered to each other, and they are replicating both of them. DNA polymerases can only attach an incoming nucleotide to the 3' hydroxyl of the DNA chain. This means that the new daughter DNA strands will grow in the 5' to 3' direction. Now, in order for this to happen, the template strand has to be in the 3 to 5' orientation. Your template strand is in the 3 to 5 orientation, the DNA polymerase can replicate it quickly and efficiently. So our antisense strand is in the perfect ready-to-go orientation. But the sense parental DNA strand, the one that's being replicated by the other DNA polymerase complex, it has a problem because it's not in the 3 to 5 orientation, it's in the 5 to 3 orientation. So there's going to be a problem in replicating both of these DNA strands. But don't worry, there is a solution to how we correct the problem. I want to stress this again. DNA polymerase III is going to move down a template strand in the 3' to 5' direction. That's the rule. A DNA polymerase heads to the 5' end of the template DNA strand. It will physically bind on to the 3' end of the RNA primer, and it will look for the 5' end and it will head that direction. In doing so, you're creating your daughter strand of DNA and it's going to grow in the 5' to 3' direction. Your zip line, the one that you were zipping down and replicating quickly and easily, the one that's in the 3 to 5' orientation, gives rise to what is called leading strand daughter DNA synthesis. You are the antisense strand. You are being replicated easily, quickly, and continuously without any problems, and this is known as leading strand DNA synthesis. So leading strand synthesis uses the antisense strand of parental DNA as the template. Your partner's DNA strand, your partner's zip line is in the wrong orientation. It is in the 5' to 3' direction, and it cannot be replicated continuously. As I said in the previous slide, your partner has to take a stretch of their DNA template, flip it, and then zip down it. So this was first discovered by a gentleman named Reggie Okazaki, who studied this strand synthesis and discovered it was done in chunks. These chunks were about 1,000 to 2,000 base pairs long. He named these chunks that have been flipped and replicated Okazaki fragments. And that discontinuous replication, that flipping and zipping of your zip line done by your partner, is called lagging strand synthesis. Lagging strand DNA synthesis uses the sense strand as its template. So how does it all happen? Well, this is the big picture, as I call it, and don't worry, there's going to be an animation for you to check out in the next slide. You and your partner, you are the alpha subunits of the DNA polymerase III. You are going to go zip lining. In other words, you're going to replicate DNA. The helicase is going to unwind the braided zip lines into 2 zip lines. So in other words, the helicase is going to create 2 separate parental DNA strands. You are going to look at your template DNA strand and see that it is in the correct 3' to 5' orientation. And you're going to say, great, I'm ready to go. A primase is going to build a primer. So there's going to be a wooden platform that is going to be built, and you're going to climb up onto that wooden platform. The zip line employer, otherwise known as the clamp loading protein, is going to take a big metal carabiner, known as the beta clamp, and is going to clamp you onto the DNA template strand. You're ready to go. Your partner has a problem, though, because they look at their DNA template and go, my zip line is in the opposite orientation. So now I have to take a stretch of about 1,000 to 2,000 base pairs, and I have to take my zip line and I have to reverse its orientation so it's in the same 3' to 5' orientation as the other zip line. Build a brand new primer wooden platform, climb up on top of it. Zip line employee takes the big carabiner's beta clamp and attaches your partner onto it. And off you both go, heading towards the 5' end of your DNA template or zip line. You're going to keep going. You have no problem. But your partner has a problem because they're going to go about 2,000 base pairs, and then they have to repeat the whole thing over again. The zip line employer has to take your partner off of the zip line. So the clamp loading protein unclamps the beta clamp, and you come off the template. You're now going to have to take about a 2,000 base pair stretch of zip line and reverse its orientation. Flip it. The primase has to build a brand new primer, so you have to build a brand new wooden platform. You have to climb onto that platform. The zip line employee has to take the carabiner beta clamp and clamp you back on, and then you head to the 5' end of that flipped section. Flip it, zip it. You got to do it again. You got to come off of the template. You have to flip the next section of template. You have to build a brand new primer. You have to attach to the 3' end of that primer, and you got to zip your way down to the 5' end of that flipped section. Over and over again, that's what your partner has to do. And they are physically tethered to you, who's just zipping down your DNA template zip line, free and easy, having a lot of fun because you don't have to do any of that flipping and zipping. Here's the animation that I talked about in the previous slide. This is the YouTube video. I want you to watch this video 3, 4, 5 times. How many times you have to watch it in order to understand what's going on. But you'll see in this video, you will see instead of flipping it like I use my terminology, they actually produce a loop. But in either way, they're doing the same thing. They are reversing the orientation of a certain section of DNA template, so that is in the ready to go 3' to 5' orientation. There are all kinds of animations to look at. Some of them are relatively simple. Some of them, like the one you just saw, actually builds it into a big picture. Other animations are actually computer simulations that show you what it probably actually looks like in a cell. So make sure you guys go to the Canvas module on DNA replication. Click on the reading because I actually embedded a few of those videos in the reading. All right, so that's DNA replication. I know your mind is swimming because it is actually a pretty complex process. But I promise you the next lecture where we get to RNA transcription is going to be a whole lot easier because all of these problems with replicating 2 nucleic acid template strands disappear when you transcribe into RNA. All right, we'll see you guys back at the next lecture. Take care, stay safe.